Welcome everyone. Thanks for coming on a Wednesday afternoon. Like Dr. Waddell said, I decided to do this talk about my experience as an undergrad going to graduate school and my path in science being an Indigenous person. I'm on Twitter. I don't know if any of you are on Twitter, but you can follow me at Theoretical Fun. I made that up when I was in grad school, so here we go. Yat Gawatsi, She Joslynn Lee Inishia, Laguna Nishla, Do Todagoja Bashashin Ado, Acoma Pueblo, Dashache, Do Ashii Dashanelli. So I introduced myself in Diné, or Navajo. So this is a way that we share our clans and find relationships with folks. So I don't know if anyone is of salt or saltwater. A few of you? All right. So we'll have to chat later if we're cousins. And Gawatsi, Joslynn Lee Shawahanutsa. This is in Karis. So I'm intertribal, coming from tribes of New Mexico. So on my Karis side, I am of Big Parrot Clan. So as I think about the space we're in today, a few colleagues in this room helped write our land acknowledgement here at Fort Lewis College. So we acknowledge the land that Fort Lewis College is situated upon is the ancestral land and territory of the Nuchu Ute people who were forcibly removed by the United States government. We also acknowledge that this land is connected to the communal and ceremonial spaces of the Jicarilla Apache, Pueblos of New Mexico, Hopi Sonoma, Hopi, and Diné Navajo nations. It's important to acknowledge this setting because the narratives of this land in this region have long been told from the dominant perspectives without full recognition of the original land stewards who continue to inhabit and connect with this land. Thank you for your attention and respect in acknowledging this important legacy. So this is important to me, and I think a lot of the folks at our school, because when I think about this area and my connection to land, these are the lands of my ancestors. So my mom grew up in a village called Mesita, and this is west of Albuquerque. Her tribal affiliation is Laguna Pueblo, also Acoma Pueblo, which is just like down the road on the highway. My dad, on the other hand, grew up in Shiprock, so he's Diné. So me coming from both backgrounds in this space, I've always wanted to come and live close to home as I've pursued my career in science. So again, I want to thank Dr. Waddell and Dr. Smith for having me a part of this speaker series. I think it's great to hear stories about how folks became interested in their current research. So today, I want to also thank everyone for coming and also celebrate Happy Indigenous Creatures Month. Sorry, that's a Reservation Dogs reference. For those that you don't know, there was some person who is on the news and they called Native American people indigenous creatures in a reference. So I think it's fitting for me to help present in this space also. Okay. So today I want to share a few ideas as I think about how did I get here, what's some advice that I can give for folks like some of you students who are in chemistry, biochemistry, but also just in STEM in general, on how you can use your cultural values to be able to make important decisions to be feel like you fit into the realm of STEM. So how do I fit into this realm of teaching and doing research at a school that serves Indigenous students? How can I put Indigenous perspective in my teaching material and be aware of all the cultural values that come in this space? So I'll start out and share a little bit about how my upbringing impacted how I perceive myself as a scientist. So I looked up the definition of what a scientist is. This is someone who is studying or has expert knowledge in the natural or physical sciences. So as I think about this definition as a child, you know, the first person who comes to mind would be my grandparents. They both carried valuable information about the spaces they live, plants, how we respect Mother Earth, and all the traditional stories that came about. So when I think about when I started with science, this is kind of like my backyard. So for those of you who are familiar, Red Valley, Arizona, this is, if you know the Shiprock Pinnacle, it's just west of this. So this is an open valley that has beautiful colors of red, and there are a lot of wild horses. So my sister took these pictures. She's a great photographer. So I spent some time, mostly during the summer. I grew up in Farmington, New Mexico, so that was a predominant community of like urban slash reservation life. But as I hung out with my grandparents and family, you know, we herded our goats and cows, and also grew different crops in this type of soil. So for me, I always wondered why some plants grew here, why we had rainforests, also, you know, why the rocks are of this color. And as I gained experience from this, I think this was a pivotal point in my life to figure out I really like science. So in my dissertation, so this is the final step as you write all your work for graduate school up, I put in a dedication to my grandparents. They didn't go to college or they didn't have the opportunity, but the knowledge from my maternal grandmother, my Shema Itza, just knowing her experience with boarding schools, being able to pass on the knowledge that, you know, the history wasn't able to cultivate that as learning. So for me, I think all the traditions that I've learned both on my Keres and Dene sides have shaped the person I am today. So this is something that I'll come back to a little bit later, but this activity of herding goats and then having her tell me what type of plants to look at, understanding how we use this medicinally to cure stomach aches and help with digestion, having that experience with science, but not realizing it was science, is something that got me really interested in chemistry. So I do get emotional about this just because when I think about how I got to be in a classroom setting, seeing scientists, they're all predominantly male, and not being able to recognize folks like my grandmother and grandpa who were doing science, who knew about the natural world and physical sciences. So trying to understand, do I fit in this, or do my values that my family carry, could we be scientists? So as I thought about, should I pursue science? In high school, I didn't really think I could do physics or chemistry just because it was not attainable. I didn't see people like me. But this will show you my age. But there's a TV series called ER. And in this instance, thinking about also uranium impacts on my family. So I've had a brain tumor, and my dad and grandfather also have cancer. So in my family, thinking about what can I do with my career and how can I start to think of a pathway in science. So I decided maybe med school could be an option because I wanted to do something that could help my family. And as I started to look at what are the pathways to science and thinking at the high school age, what I want to do with my life, I had to come to the balance of how does my culture fit in this realm. So I decided to build in this activity, and you all have whiteboards. So this is sort of leading into how I've integrated my cultural values, personal values, and how I want to hopefully have you all think about how we can be together as a community. I was up late last night watching election results, which was probably bad, but just thinking of how can we make our community work together. So with that, take about 30 seconds to think about your personal values. And as you're thinking about that, what in your daily life helps you practice or promote these values? So I don't know if we could turn the lights up a little bit to help people. Did everyone get a whiteboard? in here? Okay, so hopefully you can see, but just think about this on your own as a reflection activity. Sorry to bring down the tone with all of this, but it is emotional thinking of how I got interested in science. It's not super exciting, but I think it still has helped me today. All right. You're all very quiet. Well, it sounds like you're thinking so. Okay. So you don't have to put your full, I think, values, but introduce yourself to someone nearby or chat with a friend. Share your values with each other. And then you can either write all of them on the whiteboard. But I just want to get an idea of what everyone's values are. I have my own, and just see how we can use these as ways to guide ourselves, thinking about making our community space more positive. So, yeah, so you can chat with each other. I'll give you about 2 minutes. And if you don't want to share, that's fine, but just get an idea of what you all think about what some of your personal values are. I think I did that. Check. Okay, so that's about 4:15. Okay. Okay. So I guess your 15-second warning to come back. Okay. All right. So I'm going to go over and share sort of my overall values, but I don't know if anyone else wants to share out some of their themes. You can hold up your board. I'll try to read that far if you have volunteers who want to mention some values that they have. All right, so there's a lot of small writing, but I see things like learn, play, love, or live. Yeah. Language, yeah. And then family, hard work, exercising, empathy, education. This is testing my eyesight. So, all right. Justice, okay. Yeah. All right, so these will have some common theme. And it all depends on your cultural background. So as we get folks together, it's unique to start to see different priorities that people have. So what do you So why would I ask that, and how does it work with science? So for me, when I'm in the scope balancing my Keras and Diné values, I categorize them in 3 spaces. One is a holistic, balanced worldview. So I'll go into detail on that and how that's sort of shaped how I approach being a chemist. And then having a sense of belonging and connectedness. This is something of finding your support system in how you can succeed, exchange ideas. So science is very collaborative, but do you feel like you belong? This sort of fits into community, and being one of the few Indigenous scientists, I really look at giving back to train the next generation. So how are things that I do, not just in the research lab, but community-based, that help keep my cultural values in my daily choices? So I just want to share a few examples with you all and have you think about how you can start to propagate your values in your everyday life as a student, as a researcher, as a professor or instructor. So the example I have to start is Hajong. So we call this a wellness philosophy. This is a way that you start to think about how this belief system is not just in your thoughts and words, but how you behave and what you say. So as we think about this philosophy for Diné folks, this is sort of the moral and behavioral way and all the actions that you take to live a long, healthy life. And healthy isn't just in your physical, it's both your mental and community or interconnectedness. And as we think about how we strive to become this state of Hajong, it's really a balance that an individual takes. So as I've referenced my grandparents, they're ideal role models. When we think about our elders, these are folks who have had traditional teaching and lifetime experience. So they're on this path. They've made decisions, positive and negative, to help them reach this wellness state. So as I connect that philosophy, thinking about my cultural and social foundations, understanding how we treat our beings. So in science, we constantly tell each other we're studying these things, but I don't just see them as things. These are how I connect to Mother Earth. When we think of air and water, we take care of it. When we think about animals and plants, it's a whole interconnected system. So as I've had to learn all these various skill sets, studying proteins, looking at cells, part of me feels torn because I can harm some of these beings that I've learned about. And a lot of Western science really centralizes humans at the top. But one thing growing up is that we're not separate from the environment. All of our actions have impacts, whether it's to air, water, or other people. And then keeping cultural practices alive, having traditional foods available, sustaining on land, and thinking about how you can use nature as restorative or healing. So trying to align these values for what I do as my day-to-day life currently as a professor, this is what helps me keep going. So as I told you, I was interested in science. I decided to be a pre-med option because I thought that was probably the only entryway into science at the time. So I'm alum of Fort Lewis College, so I started here in 2002. I was a bio major initially, but I really enjoyed chemistry because there's a lot of calculations that were fun to do. Most people don't like that. But for me, this was one space that I knew there was an answer, and I could work my way through different problems. In my second year was really how I saw chemistry aligned with my culture and a lot of the traditional knowledge my grandparents had. He's no longer in the department, but Dr. Ted Bartlett was my organic chemistry professor. We had a lab that he wanted us to bring a plant in so we could extract natural dyes. I always refer back to, this is referred to as green thread, but this is a tea that my grandma would have me find these flowers. She would scold me when I picked the wrong ones. But the process that we would go to drying this plant and then use it as a medicine to help with stomach aches was one option, but also through the variety of ways to generate different dye colors. So for this organic chemistry lab with Dr. Bartlett, I was able to isolate specific dye molecules in this organic chem lab. And it wasn't too tricky because I was able to think about how my grandmother approached this process. And it's not just my grandmother. There's a lot of elders throughout the whole entire world that start to look at how we can understand dye molecules, get vibrant colors, and do our traditional weaving. So for me, at this point, I was really excited to see that organic chemistry is something my grandma was doing, but it just wasn't called organic chemistry in that context. So in the audience, I'll share Dr. Les Somerville. He was also pivotal in my career. Dr. Bartlett had mentioned this program called Mark U-Star, which is now our U-Rise program, and told me to apply to do research. I had no idea what research was. All I knew was I could do lab techniques, and it just seemed like it was fun. So again, I had wrapped up my first and second semester of organic chemistry. Dr. Somerville's lab was predominantly biochem techniques. I had not taken biochemistry, so this is very terrifying, thinking of I'm going to jump into this lab without any background. So the project we started on was investigating the metabolic processes of Acidobacterium capsulatum with high-performance liquid chromatography methods. So this is my first time working with microbes, growing them up and then trying to identify the different enzymes that are going to allow us to understand how this bacteria grows. The cool thing is I got to meet other students in our department who were also doing research. So we all traveled together to an American Chemical Society meeting and presented some of our findings that we worked on for the year and a half that I was in this lab. I did contaminate bacteria, so there was a lot of learning along the way. But in my mind, bacteria were something that didn't have a cultural taboo for me. With the U-Rise or U-Star program, you were going to do a semester here of research during the summer, and then you would go to another institution. I decided to apply to Dartmouth Med School. I wanted to figure out if I still wanted to pursue med school or stay in research because I really enjoyed my experience in Dr. Somerville's lab. Out of, I think it was 400 people that applied, 8 of us were accepted in this program, so this is pretty competitive. It was a 7-week experience in Hanover, New Hampshire. So this was more of a cell biology lab, and for me, it was a different experience because this was my first time being away from home for a long time. 7 weeks doesn't seem long, but I had not been around, on my own, thinking of being very many miles away. During this time, over the summers, I would go home and help my family with our goats and cows and planting, harvesting. So this didn't seem like a long time, but it was during the summer that it was crucial that I was available to help with my family. But talking with my parents and getting their, I wouldn't say approval, but just understanding that I'm going to miss out on all these events was really hard. But as I was able to select working with Dr. Harry Higgs, I found that I would be working with human cells. And this is the first time I had to take care of these cells, grow them, and then the main goal of this project was to look at their surface protrusions. So we have a lymphocyte cell, and it has these little finger-like protrusions. These are called microvilli. Once you go into a state of cancer, you start to form these blebs. So I got to learn some scanning electron microscopy. And this we didn't have here at the time at Fort Lewis, but I also was able to look at how we can start to understand the actin binding proteins that grow and form these blebs. So fluorescence microscopy was also something that I had never done before. So overall, this was a positive experience, but I got introduced into what it's like to be a grad student. I didn't really like working on the schedule of how cells grow, so I decided I probably don't like cell biology. But I got to see what research was like at a R1 institution and chat with grad students at various levels. The data that we generated, I was able to go to another conference, the American Society of Cell Biology, and present data from this summer. So it wasn't unusual to have undergrads present their work, but on 2 different projects. It was really exciting to have that opportunity. So I graduated in 2006. This is our class. We had lots of courses together, so we decided to sit together and put chemistry on our hats. I applied to many PhD programs. Some of them I didn't get into, and others that I did, I really didn't want to go to that program. So for me, I didn't know what type of chemistry I wanted to do. Instead of thinking about staying in the Southwest, the limited amount of jobs in biochemistry at the time weren't as accessible. So I decided to move to Massachusetts. This is where my sister lived. She went to college in Boston, and in Boston there is a booming bio or pharmaceutical, I guess, variety of things to do. So this was fun, starting as a temporary technician. Coming with the bachelor's degree, I did get my foot in the door to work with liquid chromatographers coupled to mass specs. In this instance, it was exciting because my undergrad research project was using this instrumentation. So I got to become permanent as a research associate, and this was in their discovery bioanalytical group. When you're working with million-dollar machines, this wasn't just one, we had a whole suite of them. Being 21 and just having a whole playground of instrumentation was really fun. I had an awesome boss in the drug discovery department. He told me, you're going to talk to, I think, 6 different chemists and figure out what type of chemistry you like. So Dr. Christopher Brummel was the one who introduced me to computational chemists. And this is how I got into the field of computing. So in this, with drug discovery, you'll have a disease target and a small molecule that you want to design and figure out how the interactions with the amino acids in the active site give either the enzyme the capability to halt its process or start with its enzymatic reactions. I was told at Vertex I should go to grad school because my limitations at a pharmaceutical company wouldn't be as increasing to, I think, the cool projects that I wanted to do. But one thing I faced was being a first generation. No one in my family went to grad school. So trying to think about, do I want to stay in Boston? My sister's in Boston. Everybody tells me grad school is hard. So I did talk to all my pharmaceutical company colleagues who went to grad school and asked them what are the things that I need to know and things I should avoid. The other thing I had to think about were my family roles. I had been at the job for about a year when I was thinking about applying to grad school and talking to my parents when I want to move home, because I always wanted to come back here. And one thing that I always had to have these discussions, because in my family we have a history of boarding school impact. So my grandmothers both attended Sherman Institute, and in that experience, the time away when they were 17 changed them a little bit. So family really wanting you to return home. It's not just a thing, it's really integrated with boarding school history. So I decided if I'm doing computational chemistry, I can usually work from anywhere. In this context, I just need the internet and a computer. So that's where I decided to choose my grad school topic of computational chemistry. And I was kind of tired of working with specimens, as scientists call them. Human cells, those come from an individual. There's a part of them. In my preclinical trials for working in drug discovery, we used a lot of model organism tissue. So I was working a lot with mice, rat, guinea pig, monkey. And I just didn't like that in the space I was going into. So computing seemed like a safe area for me and my cultural values to have a safe space to learn. So I applied to Northeastern University and I decided to stay in Boston. I really looked at the molecular modeling topics that Dr. Mary Jo Ondrican at the school had projects on. She was one of the first Indigenous chemists I ever met, so it was really cool to have her as a role model. She is female in one of the fields that was predominantly dominated by males at the time. So I learned methods to predict protein function from 3D structure, and this was not just biochemistry, it was math, computer science, and learning computing. Going into a graduate level space, there is a lot I had to learn, but it was really exciting for me. Our role, or our main goal, was to look at how we can understand evolutionary relationships of enzyme active sites just by studying all the available structural information that was coming out at the time. So this was some of the data files I had to work with. For me, looking at this for the first time, I was like, it's just all these letters and numbers. But as you start to look at each line, you'll see there's some C's, N's, O's. So these are some of your atoms. And if you go over a few columns, you start to see negative values. And this is what we call a protein coordinate file. So learning about what makes up some of the structural information we have of proteins, each of these carbons, nitrogens, start to be visualized in a molecular modeling software. So we can actually study all the bonds that make up a ligand that we see here bound into this protein site. So we see these alpha helice ribbon-like structures, but those actually represent amino acids that make up a protein. So our group was trying to figure out, could we identify these protein active sites that interact with ligands and then try to decipher some of the function that comes about. So some of the more mathematical interactions with proteins on the biochem side is we looked at the atoms that were within a distance of each other, and these amino acids we represented as a sphere. And we took these triangulations through a series of programs that we collaborated with folks at our computer science department at Northeastern. And here we gave weights at which amino acids were of high rank value. We had a parameter on what we would say if this was like an aspartate. It would be heavily weighed, and then we'd look at surrounding amino acids nearby a certain distance. So what we wanted to do was take all these unknown structures and see if we could create these ball and stick maps, overlay them on each other, and see if we could predict what type of function they could have. So my role was, how do we automate this process? How do we start to study one superfamily of enzymes and then be able to relate that back to our question of how can we understand more of the evolutionary relationships. You have proteins that are in plants, you have proteins that are in humans, so how similar or dissimilar can they be? So it was a really exciting project. I didn't envision myself becoming a computational chemist or have that training. The only downsides of this that came out was being one of the few Indigenous folks in my research group, my department. The positive sides were that I could go abroad and present my research. So that was really exciting, and I couldn't have done it without my parents and all their support and my family. In 2014, there were 6 American Indian and Alaskan Native folks that received their PhD in chemistry. I was one of them, and I knew 4 of the other people. We couldn't figure out who the other 2 were, so we're always trying to find out who's there. So at this time, thinking about where do I go next in grad school, I really wanted to become a professor. So I tried to keep an eye on if there's anyone retiring at Fort Lewis, because this was the ideal location I wanted to be, and then what type of training I needed to do. So most of the time you do a postdoc experience, and I wanted to go to a place that had more Indigenous population. So I decided to do my training at University of Minnesota in Duluth. I also wanted the small town feel again. Dr. Matthew Slattery had just joined the med school. He was doing work that was interfacing more computational biology, but also needed someone with wet lab experience. So even though I was outside of the lab for 6 years doing my computational training, I was still able to pour gels and because that's, I think, one of the trickier things to do is pour an SDS page gel. I was able to grow cells again. This is when we're trying to purify proteins in some of these cells. So half of my time was in the computer lab, the other half was in the wet lab. So that experience was interesting because when you're a postdoc and you're trying to get a tenure track position, publishing became a priority. So I got a few publications out with a collaboration with another postdoc at Johns Hopkins, Raj here. We actually only communicated over telephone at the time just because conference calls, we didn't really have Zoom. We had Skype, but it was kind of wonky at the time. But going to the same conference a few years later is really cool to meet each other on all the projects we collaborated on. But as I was in my role as a postdoc, even though I wasn't faculty, having to have a voice on campus climate. So this was the pivotal time that I found out I'd be doing way more service if I became a faculty member. Having to dismiss stereotypes that students had of Indigenous Alaska Native folks, having students who wanted a presence on campus. So American Indian Society of Engineers and Scientists is a group of students. We had maybe 8 at the time who were in our side of the chemistry and med school. We had never held a round dance on our campus, so I was able to connect with folks to give us support on how we could invite the community to campus. So this was something that I think the students really enjoyed, just having them be themselves on campus instead of just being a student that was constantly stereotyped. This is one realm of my local community on campus, but also getting to know the folks or where these students come from. I had not really met a lot of Anishinaabe folks from northern Minnesota, but one thing, Duluth is known for missing and murdered Indigenous people. So this was one thing that I had to keep in the back of my mind, even though I was highly educated. This could still be a possibility because this was happening in the community. I also met an awesome group of Indigenous runners. So Sarah Agaton Howes started the Koi Pack, and she's the owner of Heartberry. But we did some 50Ks together, so it was a lot of fun to get outside and sort of take a break from all the stress. Learning about traditional foods of the local community, getting invited to all the opportunities to know where these students are coming from and ways to connect and support them. Most faculty weren't doing this at University of Minnesota Duluth, so having to step up as a role as a postdoc was a big ask, but I had students who wanted that support. So I had to have a career check-in. I know we're coming on time, and trying to think about how do I fit as a scientist? Do I want to pursue this path? And then thinking about the traits of Hajo. So coming back to this, reminding myself what Dene elders have. So there's a paper that I linked here that kind of summarizes these if you want to know a little bit more. But these are the traits: being respectful and thoughtful, thinking positively in your thought, speech, and behaviors, being supportive, being a good and attentive listener, being creative, being deliberate in action. I think I saw empathetic somewhere on someone's board, being patient. So as I reflect on these values, these are describing both a teacher and a scientist. So as I think about my values, all my day-to-day decisions, it really comes back to this mindset. So I decided, let's try to get a job at Fort Lewis, and that did happen, spoiler alert. But I started in 2019 in the chemistry and biochemistry department. I was one of the first female Indigenous assistant professor to join the department. So this was kind of shocking, but thinking about our diverse population here, very important to have that voice. And as I was looking at this job, thinking of being a tenure track professor, what type of research do I want to do? So this comes back to the idea of community. So I do community on the micro scale, so I am still studying microbes. I enjoyed my time in Dr. Somerville's lab. And then I want to build a sense of community here on campus at Fort Lewis so everyone can see themselves as scientists and have role models to learn from. So I'll do a few slides on my research project, but while I was not living here at the time, there was the Gold King spill in 2015. This released a lot of metals and polluted the riverways of my hometown in Farmington, downstream into Utah, and obviously here in Colorado. So again, the Animas River flows down from the location of Silverton, and the Gold King mine is a creek just upstream. And all of that traveled down over the course of 2 weeks to end up in Lake Powell. The main thing I was concerned of are all the folks who utilize the water for agriculture and livestock. So I wanted to know how we can start to understand the resiliency of our rivers. So when we start to think about not just things we see visually, microbes are doing a lot of cool things that help us live. And again, coming culturally, I don't want to work with human cell lines. I'd rather not work with animals and plants. So microbes seem like a safe space for me culturally. They're doing really crucial nutrient and elemental cycling that's important for us and our other beings to live. So when we think about microbes, they're doing also some really cool metals chemistry. So if we're thinking about acid mine drainage, there are going to be a specific type of bacteria that are going to be the ones that uptake metal. A lot of their metabolic potential, ecological functions, relationships in their small communities isn't as well studied. So I'm looking at a specific group of iron oxidizers because they can also uptake different metals. And if you go back to what occurred in the Gold King mine spill, we do have contaminations of these metals in our waterways. So for the questions I'm asking, I'm trying to think of, is there a way to link bioremediation? How can we use some of these microbes to clean up our water? And is there a less toxic way to do this? Because right now, the methods that they're doing at Gold King Mine aren't sustainable. So I'm trying to look at what is currently there, cleaning up some of the water. Okay. So we've all encountered the pandemic. So starting in 2019, I didn't have a summer research group. I also had to have a brain tumor removed. And in this case, I wasn't able to do research until 2021 with students. I got students in the field. We grew up some unique microbes. And this was my research group. We were all Indigenous. So for me, it was super exciting to have students that come from a similar background interested in how we could help Mother Earth. So this is one aspect of my research lab and thinking about communities and effects that we have with them on the micro scale. Other things I'm doing on campus are starting the Colorado Outdoor Equity Group. So I've got funding to take students out to explore spaces. So if you're interested with that, building community on campus is something I do. Training the next generation of scientists. So I'm on a genomic data science research community. We're looking at how schools that are not R1 research intensive, but people coming from tribal colleges, historical Black colleges, Hispanic serving institutions, and community colleges, how we can get them to be in the realm of genomics research, computing, and where do we start with that? The other thing I'm starting to explore is how we can teach folks about Indigenous data sovereignty. This is something that I haven't had formal training in, but it does complement into the types of opportunities you get working with data. So I'm working with folks at University of Arizona on how we can prepare teaching materials so that we can expand them out to public health departments. So this is sort of my non-lab research, but it's really fun collaborating with folks like Dr. Stephanie Carroll, and we're excited to share some of this teaching material. Um, Other things I'll just highlight briefly is not just starting here at Fort Lewis, but thinking of high school level. So I'm part of this co-directing of, I guess I need to point this way, culturally and academic research experience. So thinking about how we can, oh no, I'm almost done. Maybe this is telling me to hurry up. There's food outside. There. So we wrote a chemistry and culture chemistry focused lab resource book. So thinking about how you can integrate your indigenous culture in a lot of the chemistry aspects that you do. And this is all taught on Zoom during the pandemic, and we ended up keeping it through just because we know there are students who are at remote locations on tribal lands who want to have access to chemistry training but may not have this. So we developed some cooking kitchen experiments so that students can do this in their home. The next thing is, again, getting computing to high school students. So we've built these kits of Raspberry Pis and trying to get high school teachers to be able to teach their students about air quality issues by writing code in Python and having these smaller, cheaper computers to be able to look at their local data sets. And lastly, this is sort of my child, I would say. So I started this monitoring environmental microbes. How can I train other people to do research studying microbes? So this is a workshop that's held annually in May. We have funding from Anschutz, and we get students to process microbiome data. This is also having impact on Indigenous values as we start to be land stewards, bringing other scientists who are able to give insight into how we can help study and improve our environmental systems. And the exciting part is I submitted a paper on this workshop, so that's really fun to share how we can get these types of experiences for students who identify as Native American or underrepresented opportunities at their school too.